Hey guys, so today is the last lesson of the genetics unit. We're going to be talking about gene linkage and crossing over, which we have already touched on before. And then the one new thing that we're going to be doing that you will definitely see a question on the unit test is creating gene maps. So we're going to start by just reviewing what gene linkage is. We're going to briefly talk about how Punnett squares are affected by linked genes, so you are not going to have to do any linked gene Punnett squares on the test. Then we're going to briefly talk about how crossing over affects linked genes. We're going to calculate the percentage of crossing over, and then the most important part, creating those chromosome maps. So the chromosomal theory of inheritance says that genes are carried on chromosomes, which we already knew, that chromosomes will segregate independently into gametes, so that they'll go to the opposite sides, and they'll be sorted not based on maternal or paternal, but at random. And this theory is based on the movement of chromosomes that follows the patterns that were predicted by Mendel when he was doing all those experiments on his pea plants. In 1902, a biologist called Walter Sutton and another biologist called Theodore Boveri both saw that the segregation of homologous chromosomes was happening during meiosis, so that did support everything that Mendel was seeing with his pea plants. Those two scientists determined that alleles are located on chromosomes, which again are the different options for genes that are on chromosomes. Sutton then predicted that when two alleles for two different genes are on the same chromosome, they won't assort independently. So those genes won't be just randomly shuffled into different gametes. Because they're on the same chromosome, those two different alleles would be inherited together. Those genes are what we call linked genes. Like I said, they're usually inherited together. When Thomas Morgan was experimenting with his fruit flies, he noticed that gray bodies and straight wings were expressed together, and black bodies and curled wings were expressed together. So he determined based on that the traits for body color and for wing shape must be on the same chromosome. Those genes must be linked. Morgan then looked for other traits that always appeared together like that in order to see which traits were likely on the same chromosome. So yeah, he noticed that those traits must not be getting separated during meiosis because they're on the same chromosome. Just a reminder, this was also the guy who came up with the idea of sex-linked traits. So if you're doing a normal Punnett square and you have two individuals, and you're looking at two traits for each of them, and they're both fully heterozygous, you'll always get a 9:3:3:1 ratio. If those genes are linked, though, in this case, gene A and gene B, if those genes are linked, you're not going to get that 9:3:3:1 ratio because the alleles aren't going to be separated into gametes the way that we would predict they would be. Normally, when we're creating gametes in a scenario like this, you use like the foiling method. So your gametes would be like capital A, capital B, and capital A, lowercase b, and lowercase a, capital B, and lowercase a, lowercase b. If the genes are linked, they're not going to separate that way. So if you were doing a cross between these two fully heterozygous individuals, but the dominant alleles are linked and the recessive alleles are linked, then you only have two possible gametes instead of four. So you would end up with this kind of scenario instead. You can only have either the dominant alleles together or the recessive alleles together. You can't have one dominant and one recessive. Again, this is just kind of like for your information, but you're not going to be tested on this. We talked about crossing over when we were going over meiosis. Just a reminder, crossing over happens in prophase 1 of meiosis. It is when you have homologous chromosomes pair up with each other, they form that tetrad, and then two of the non-homologous sister chromatids, like cross over and swap some alleles with each other. Crossing over is the only process that can separate linked genes, that can separate genes that are normally on the same chromosome. So yeah, the more closely together two genes are on a chromosome, the less likely they are to be separated. The further apart they are on a chromosome, the more likely it is that one will get moved to a different chromosome during crossing over and the other one won't. When you have linked genes on a chromosome, sometimes we do call them a linkage group, especially if there's more than just two linked genes. Yeah, crossing over is random, so it can occur at any point on the sister chromatids, except right beside the centromere, which is the thing that holds the sister chromatids together. The frequency or the probability of crossing over, we can state as a percent. And again, that all comes down to how closely the two genes are on the chromosome. So the crossover percentage is sometimes called the recombination frequency, and it's just the frequency that crossing over will happen between two genes. The closer together the genes are on the chromosome, the lower the crossover or recombination percentage or frequency will be. Again, because it's less likely that those genes will get swapped away from each other if they're right next to each other. The further apart they are, the greater the recombination frequency, the greater it is that this one allele will get switched to a different chromosome. We can calculate crossover percentage by doing the number of recombinations divided by the number of offspring, and then of course multiplied by 100 into a percent. So in fruit flies, just an example, in fruit flies it says that there are gray bodies and straight wings that are linked, black bodies and curved wings are linked. Again, it was Morgan who taught us that. If in the F1 generation of fruit flies, there were 220 with gray and straight wings, 63 that were black with curved wings, 5 that were gray with curved wings, and 7 that were black with straight wings. So when we're determining crossover percentage, we're taking the number of recombinations divided by the total. The number of recombinations are the number of individuals that their linked genes aren't showing up together. So they have a weird combination of traits, essentially. So we know that we should have gray body with straight wings. As soon as we have a gray body with a curved wing, that is a recombination that's occurred. We should have black body with curved wings. As soon as we have black body with straight wings, that's a recombination that's occurred. So if we look at these numbers here, these 5 individuals that were gray with curved wings and the 7 who were black with straight wings, both of those are recombinations. So our number of recombination is just 7 plus 5. And then for the total number of individuals, we just have to add up everybody. So 220 plus 63 plus 5 plus 7. So what you should end up with, if I just do the math really quick here, is 5 plus 7, which is 12, divided by 220 plus 63 plus 5 plus 7, so 295. So the 12 recombinant individuals divided by the total 295 gives us 0.04, and then we multiply by 100 to get 4%. So our crossover percentage is 4%. Again, all that I'm doing there is taking the number of recombinations, which is the 12 plus the 5 divided by the total. So that's the 220 plus the 63 plus the 5 plus the 7. We divide and then multiply by 100. Okay, that's all that this slide is showing as well. So our recombinant types are the 5 plus the 7, the total, add everyone together, divide, and get 4%. Okay, this is the most important part, chromosome mapping. So with chromosome mapping, all you're doing is you're using crossover percentages in order to determine how far apart different genes are on a chromosome. The higher the percentage, the further apart they will be. And so you just kind of create a straight line and draw where different chromosomes will be on that based on their recombination frequencies or crossover percentages. So we'll just run through a quick example here. So we have 3 different genes in this case, the eye color gene, body color gene, and wing length gene. We represent each of them with, in this case, 1 letter, but sometimes it's 2 letters. And then you have to be giving some crossover percentages. So in this case, you're told that the E and B genes have a crossover percentage of 5%, so they're fairly close together. The E and W genes have a crossover percent of 30%, and the B and W genes have a crossover percentage of 25%. Each percentage we call 1 chromosome map unit, sometimes called a little c capital M, other times called an MU for map unit. So what this is really saying is that the E and B are 5 centimeters chromosome map units apart, E and W are 30 map units apart, and B and W are 25 map units apart. So the way that we would draw this, I've kind of drawn like a weird chromosome shape here, but normally you would just draw it as like a straight line, and then show where they fall relative to each other. So you might just have like a straight line, and then you would have E on one end, and then you would have your B, and then on the other end you would have your W. So something like that. Also, it doesn't matter if you write the letters on the top or the bottom. I don't know why I did some of them on the top and some of them on the bottom, but that's all it would look like. Okay, I will be giving you some practice questions about chromosome mapping. So go practice those and let me know if you have any questions.